Complement depletion is a fast, effective, and inexpensive method for purifying immune cell populations. Complement depletion begins when monoclonal IgM or IgG antibodies are added to a cell population which target a specific cell type. In this example, we will target T cells for depletion. The complement system is then added, initiating a proteolytic cascade which results in T cell death. The cell population is then centrifuged, washed, and filtered to eliminate dead cells and debris. This complement depletion method typically results in the elimination of 95 to 100% of the targeted cell population.
Hi, I'm Bonnie Diddle. I'm an investigator at the Blood Research Institute of Blood Center of Wisconsin, located in Milwaukee. Today I'm going to show you the technique complement depletion, a fast and inexpensive method for the depletion of specific cell populations. We use this technique in my laboratory to highly enrich immune cell populations that then we use for a number of bioassays, including those involving cell activation and protein expression analysis. So let's get started.
Before beginning the experiment, prepare 0.5 or 1 milliliter aliquots of rabbit complement and store in either a -20 or -80 degree freezer. Be sure to titrate the complement to an optimal dilution between 1 to 10 and 1 to 20. An excellent commercial source for complement that we routinely use is from Palfrey's Biologicals. For this procedure, mouse and rat monoclonal antibodies are generally effective at complement activation, but not all antibodies will be suitable. IgM antibodies are generally the most efficient for complement-induced cell lysis. IgG antibodies are the second most efficient and are typically used because most monoclonal antibodies are IgG. Titrate each antibody for optimal use in a cell density of 10 to the 7th cells per milliliter, which is determined empirically. Most antibodies are used in the range of 0.1 to 10 micrograms per milliliter. For the depletion of splenic T cells, we use a monoclonal antibody specific for Thy1. To begin complement depletion, dilute the cells in cell culture medium containing 5% heat-inactivated fetal calf serum so that the starting cell population is 2 times 10 to the 7th cells per milliliter. For example, 5 times 10 to the 7th cells should be diluted in 2.5 milliliters of medium. This volume of cells is 1/2 of the final volume and hence twice the concentration of cells used during depletion, which will be 1 times 10 to the 7th cells per milliliter. At this time, save up to 1 times 10 to the 6th cells so that the depletion efficiency can be quantified at the end of the experiment. In addition, thaw the required amount of complement at room temperature. We have found that a dilution of 1 to 15 is appropriate when using Palfrey's Biologicals rabbit complement. For 5 times 10 to the 7th cells, 1.5 milliliter frozen aliquot is sufficient. Now add the appropriate amount of antibody to the cell suspension and mix. In order to sterilize the complement, use a low protein binding syringe tip filter. Fill the syringe with 1 to 2 milliliters of medium before adding the complement. For 5 times 10 to the 7 cells, we will need 340 milliliters of complement for a 1 to 15 dilution. Then, filter the complement directly into the tube containing the cells and antibody. Now, adjust the final volume in the tube so that the cells are at a concentration of 1 times 10 to the 7th cells per milliliter. Next, allow the cells to incubate at 37 degrees Celsius in a water bath for 1 hour. Remember to mix the cells every 15 minutes by gentle inversion. After the incubation, centrifuge the cells at a speed that will not affect viability. Then, back in the hood, discard the supernatant. Next, wash the cells twice with 10 milliliters of medium. Notice how the cell debris collects with some sticking to the sides of the polystyrene tubes. After the last wash, filter the cells through a 70-micron sterile nylon cell strainer. Finally, check the cell purity by comparing the cell population of interest before and after depletion. flow cytometry and immunohistochemistry are preferred methods used to determine the cell purity prior to experimentation. When using a monoclonal antibody that's highly efficient at activating complement, the first round of complement depletion will deplete the cells by more than 95%. As an example, using flow cytometry to measure the percentage of alpha-beta T cells in a mouse splenocyte cell preparation, we found they contained 32.2% alpha-beta T cells. Following complement depletion with anti-Thy1, which is expressed by all T cells but not by other leukocytes, the TCR-beta population was reduced by 95% to a final concentration of 1.56%.
I've just shown you a fast and effective method of depleting specific cell populations using antigen-specific antibodies in complement depletion. When doing this procedure, remember to test each antibody for effectiveness and to titrate your complement in a pilot experiment before you begin. That's it. Thank you for watching and good luck with your experiment.